within a subject there are 4 topics. One is protein synthesis, okay, other is vitamins and minerals, other is organ function test, and the fourth thing is the tools of biochemistry. There are 3 tools of biochemistry. One is restriction, fragment length polymorphism, PCR, radioimmunoassay. The fourth thing is actually a forensic medicine topic, DNA fingerprinting. They're asking a lot of questions on DNA fingerprinting principles and steps. So DNA fingerprinting is an indirect question wherein, because there was a rape case happened around in 1980. They wanted to catch the suspect, right? They used RFLP technique. Based on that, they matched DNA sample at the crime scene with the DNA sample of the suspect. Okay, that's how the case is solved. So this is the origin of DNA fingerprinting. So DNA fingerprinting is not a separate entity. DNA fingerprinting uses reflex in the past, PCR now, because PCR is more sensitive, less sample is required, sophisticated. Yes, it is costly. Yes, there is risk of contamination. But even in a decomposed sample also, PCR yields better DNA analysis than reflips. Otherwise, both reflips and PCR are part of DNA fingerprinting. So forensic medicine part is done. So if you read these 3 topics, you can apply this knowledge in the forensic medicine part integration, that is DNA fingerprinting. If they ask you what is the principle of DNA fingerprinting. And also, surprisingly, the inventor of reflip, that is Alex Jeffrey, is the same one who invented DNA fingerprinting, because reflip was the first technique which is used for DNA fingerprinting. Right? So what kind of questions are asking in reflip, PCR, radioimmunoassay? These tools of biochemistry, they're asking 3 things. One, what is the principle? The other is steps, and what are the applications? Principle, steps, and applications. There is one more area I had seen a lot of questions coming, that is variants of, special focus on variants of PCR. With COVID in 2020, you might know what is reverse transcriptase PCR or RT-PCR, which is done more common. And also what is real-time PCR. What is its advantage? There are new things which can be asked, like unasked version, nested PCR and inverse PCR. We try to cover in the regular course. Apart from that, multiplex PCR, hot start PCR are trending nowadays. But I don't want you to focus more on these things. But at least nested PCR and inverse PCR can be asked in the exam. Now, when you see nested PCR, instead of using one primer in PCR, we use 2 pairs of primers. And also nested PCR and also inverse PCR is a mix of reflips plus PCR, because in inverse PCR we first use restriction fragment polymorphism, we create a new DNA, spherical DNA or circular DNA, then we apply specific reflip, then we do that investigation, generate a linear DNA followed by PCR. So it is inverse PCR. Whatever is it, the first thing is to write introduction to these questions, that is radioimmunoassay or PCR or reflips or DNA fingerprinting. If you want to write introduction, you have to start with an inventor, and I could see most of the students are not giving proper introduction to these questions. So if they ask you what is radioimmunoassay, you have to say that it is invented by Solomon Burson and Rosalind Yalow in 1959, and they were awarded Nobel Prize in 1997. Please memorize it, because you could see in 2019 they asked a question. In 1997, Rosalind Yalow was awarded Nobel Prize in Medicine for inventing an important technique which was considered a breakthrough in endocrinology. What is this technique? Describe briefly the principle behind this technique. Even if you know PCR, if you do not know that PCR is invented by Caribe Mullis in 1980 and Nobel Prize was got in 1993, if they ask you what are the steps and principles of a technique which is invented by Caribe Mullis in 1980 for which he was awarded Nobel Prize in 1993, if you don't know Caribe Mullis was the inventor of PCR, you will not get the 10 marks. Right? So these are the 3 scientists which you should memorize. Caribe Mullis for PCR, Alex Jeffrey for reflip and fingerprinting. So your introduction is done for these questions. And one more area people are struggling is the applications part, because 3 of these methods have similar applications. If you take reflip and PCR, we're not doing reflip now because reflip has a lot of disadvantages. PCR has only one disadvantage. What is that? PCR has only one disadvantage, that is there is risk of DNA sample contamination, because this test is so sensitive, there is risk of contamination. Apart from that, the applications are similar, but when you write answer for these questions, you have to highlight the major application. Reflip was used for mainly forensic identification for DNA fingerprinting technique, and also mainly paternity dispute solving. So focus more in the lines of genomic sampling or genomic sequence identification or species identification. RefLip has more involvement in identification of a genome, not multiplication of a genome or making million copies of genome or diagnosis of disease. So PCR is mainly used for diagnosis of disease. The COVID-19, we used it, right? RT-PCR for quicker results, very faster results. Even with simple sample, you can identify any genetic disease or an infectious disease or a cancer, whatever it is, you can use PCR for that. PCR has like millions of applications, but major focus is human diseases, not genomic studies or not epidemiological studies or not crime scene solving and all. Yes, we do PCR nowadays, though it is costly, but RefLip was invented for forensic identification and paternity testing. So you need this distinction while mentioning the applications. But on top of that, let me tell you, RefLip and PCR both are DNA fingerprinting techniques. Both have almost similar applications, but PCR has more advantages than RefLip. You have to highlight this point. RIA, radioimmunoassay, is entirely a different pen. It is highly sensitive. It can detect even small quantities of biological fluids like TSH, ACTH, enzymes, hormones in the body, and also drug levels, therapeutic drug monitoring. You take lithium. Lithium is toxic if it exceeds the therapeutic drug levels, right? So to detect that, we do radioimmunoassay study. See, PCR and RIA has come recently from 2020. 21, 22, 23, it's got repeated. So if you want to choose one topic which is expected, because is RefLip, because long back in 2019 they asked RefLip, it was not repeated. So they might ask you this time, what are the steps, principles and therapeutic uses of RefLip. When I say principles and steps, students are getting confused. Principle is different, steps are different. Steps, you know, the multiple steps you need to revise. But principle part, PCR, the principle is enzymatic replication of DNA. We use DNA polymerase to replicate one DNA into million copies of DNA. That is PCR. So how it is done? We have denaturation, annealing, renaturation. So these are the steps. Don't write denaturation, annealing, renaturation in the principle part. Similarly, radioimmunoassay. Yes, there are three important principles. One which is based on immune reaction, second is competitive displacement reaction, and other is radio emission. RefLip also, the principle is we use restriction digest principle. We use an enzyme called restriction enzyme to break the DNA into multiple copies of different length. So you compare this different lengths with some length to identify or differentiate a species. That's it. So this is the principle of RefLip. So with this simple discussion, we're done the whole crux of the topic, how to start an introduction and how to differentiate applications of different topics and why name of the scientist is important to identify a particular question, what is the body of the question. And the unasked version, they might ask you nested PCR or inverse PCR, which are variants of PCR. Apart from that, I don't think you will be getting any surprise or bouncer question from this particular topic.